As soon as you read biology in high school, you learn that the genes code for proteins which form via an intermediate molecule RNA. But then we now know that more number of genes code for RNA that never forms proteins. This RNA must be then doing something. The past few decades have shown that this RNA that is not coding for protein or non-coding RNA plays an important role in deciding the formation of other proteins, or as to say whether and when a specific gene is active to form a protein. When we talk about visible features and say that genes might be responsible, then mostly it is the protein that is being formed from the gene that is responsible. But that may not be true in the case of butterfly wing colors. So who doesn't like looking at butterflies flapping their beautiful wings as they move from one flower to the next? Have you ever wondered what makes the butterfly have such colorful wings? By studying white butterflies, scientists found that it is not the protein but an RNA that decides whether the color pigments will form or not in the butterfly. This RNA molecule is a long non-coding RNA or LNC RNA. LNC RNAs are known to be involved in many functions in the body and play important roles in the development of many diseases. But the current study is the first example where a visible trait is getting affected because of an LNC RNA. Tail of the tail. With the myriad functions the tail serves for organisms that have one, it is still a mystery as to why and how the common ancestor of apes and humans lost their tail. One of the hypotheses for why our ancestor lost the tail is that the bipedal walking or walking on two limbs was easier without a tail. But a lot of animals have a tail and can still walk on two limbs. Certain AI models for robotics have even shown that a tail actually helps in stability while moving on two limbs. Although we do not know the why, but a group of scientists may have found the how of the tail was lost. Researchers in New York studied one of the genes, TBXG, which is involved in tail development in various species including mice and cats. They found that in apes, including humans, the TBXG gene is altered in a way that forms a shorter RNA as compared to the RNA that gets formed from TBXG gene in other primates that have a tail. So the code of DNA is first transcribed into an RNA molecule which further forms a protein. So this shorter RNA being formed means a shorter protein was being formed in apes, which could be the reason for the loss of the tail. The scientists then introduced this altered TBXG gene of apes in mice to test whether the mice lost their tail. And they indeed lose their tails. I wonder now whether these mice can be trained to walk on two limbs like humans. Beyond the role of glue, half of the cells in the brain are not neurons but glia. The glia cells have been known to be the supporting cells of neurons till now. An increasing number of studies are now showing that glia are much more than being the glue for neurons. Repetitive behavior or doing an action again and again is present in disease conditions like moving objects around to be in a specific order as seen in Obsessive Compulsive Disorder, repeated movement of limbs in Huntington's and Autism. A particular protein was found to be reduced in the brain of individuals with these conditions. In a surprising finding, the scientists found that this protein forms not in neurons but astrocytes, which is a type of glial cell. When they removed this protein from mice, then repetitive behavior in the form of excessive self-grooming, excessive licking of water source, or excessive exploring of a familiar object stopped. Although they still need to find how astrocytes are controlling neurons and thus the repetitive behavior, but this finding may soon lead to a better understanding of diseases like OCD and autism.